often hear someone's loss is someone else's gain, and this is truly applicable to the field of neuroscience, where we study the functions of brain based on what functions are lost when person loses that part of brain, either as a part of surgery or due to some injury or due to something like a stroke. So this is a two-part series where in this video we will cover the development of brain and parts of brain, and in next video we will see the imaging of brain and functions of the brain. So if we see an embryo developing in its mother's womb, early in its week of development, a neural tube is developed from ectoderm. This is a hollow tube. The outer cells of this tube will form the nervous tissue and the central portion will develop into ventricular system. Around 3 weeks of development, the head portion of this tube begins to show 3 bulges. These bulges have ventricles developing within them, which we will discuss later on. As these bulges are hollow inside, they are called as primary vesicles of brain. So these are some old terminologies where the word encephalon is used to represent brain. So first, towards the most head end, we have prosencephalon, which is forebrain. Then we have mesencephalon, midbrain, and towards the tail end, we have rhombencephalon called as hindbrain, which continues as spinal cord. Some places you might find head end referred as rostral end and tail end referred as caudal end. These primary vesicles further divides, folds, and develops into secondary brain vesicles. Prosencephalon divides into telencephalon present rostrally and diencephalon present caudally. Similarly, rhombencephalon divides into metencephalon rostrally and myelencephalon caudally. However, mesencephalon does not divide. These secondary brain vesicles further divides and grows into an adult brain structure. Here, interesting aspect of human brain development can be seen, where the telencephalon grows much faster than rest of the vesicle, and thus soon it starts covering the diencephalon and mesencephalon. Because of this differential growth in adult brain, the structures developing from diencephalon and mesencephalon are present in between the two cerebral hemispheres, which develops from telencephalon. So as we saw, the prosencephalon divides into telencephalon, which develops into two cerebral hemispheres. Then we have diencephalon, which forms thalamus, hypothalamus, and epithalamus. Mesencephalon develops into midbrain. Then the most rostral part, the rhombencephalon, divides into metencephalon, which develops into pons, and posterior aspect forms the cerebellum. The rhombencephalon also forms myelencephalon, which further develops into medulla oblongata. So we saw how these bulges develop into adult brain parts. Now let's come to the ventricular system, which is developing within the brain. Within each of the primary vesicles, we have its corresponding ventricles. One for prosencephalon, one for mesencephalon, and one for rhombencephalon. As the forebrain develops into two cerebral hemispheres, each hemisphere has its own ventricle. Like this, we get total of 4 ventricles. So finally, we have 2 lateral ventricles, one for each cerebral hemisphere. They drain into the 3rd ventricle present within diencephalon through opening called as foramen of Monro. Now this 3rd ventricle drains into the 4th ventricle present in metencephalon via aqueduct of Sylvius, which passes through mesencephalon. The 4th ventricle continues down further into spinal cord as central canal. The 4th ventricle connects to the subarachnoid space via 2 lateral foramen of Luschka and 1 central foramen of Magendie. So guys, we have studied how a human brain develops and how ventricular system is formed. Now let's see what are the different layers which protects the brain from external injuries. Here the outermost layer is formed by scalp, followed by the bony skull, which protects the brain from external physical forces, especially the compressive forces and various various physical injuries. If we dissect through the skull, we will encounter 3 layers of membrane. The outermost membrane is called as dura mater, followed by arachnoid matter, and the most deeper layer is the pia mater, which is attached to the surface of brain. If we look at these meninges more closely, we find the dura mater has 2 parts. The outer part, which is actually attached to the skull itself, is called as endosteal dura. The second layer, which is towards the brain, is called as meningeal dura. This dura mater is a very hard covering, and sandwiched between its 2 layers are your dural venous sinuses. Beneath the dura mater, there is a spider web like structure. It is called as arachnoid matter. Here the term arachnoid means spider. Between arachnoid matter and pia matter, we have a subarachnoid space where circulates a fluid called as cerebrospinal fluid, or simply CSF, which is an ultrafiltrate of blood, and it flows through ventricles and subarachnoid space. Another interesting aspect of this arachnoid membrane are its projections into the dural venous sinuses. These are present in some places and they are called as arachnoid villi. These are the places where the CSF is absorbed back into the blood. So you can see this thin pinkish membrane which is following the contour of brain. It is called as pia mater. The outer area of brain is grey in color and is called as grey matter, and inner portion is white and called as white matter. The cortex is folded into multiple elevations and depressions. These elevations are called as gyri and the depressions are called as sulci. Let's get acquainted to the 3D structure of brain by looking at it from different angles. So here you can see how a brain appears from side. That is what we called as a lateral view. Similarly, this is the front view and this is the top view. So guys, here you can see the cerebrum is divided into left and right cerebral hemisphere by a deep groove called as longitudinal fissure. This passes through the midline, or what we called as median plane. The 2 hemispheres are not completely separated, rather they are connected at corpus callosum. As we previously saw, there are many folds on cerebral cortex, which we called as sulci and gyri. The depression sulci, some of them are very deep and they divide the cerebral cortex into cerebral lobes. Here the most front end, what we find is called as frontal lobe. Behind that is your parietal lobe, and the sulcus which divides these 2 lobes is called as central sulcus. Now the most posterior lobe of brain is called as occipital lobe, and it is divided from parietal lobe by parieto-occipital sulcus. There is one more lobe of brain which you do not see in this view, but it can be seen from the side or lateral view. It is called as temporal lobe. It is separated from the frontal and parietal lobe via lateral sulcus. The anatomical differentiation between occipital lobe and temporal lobe is an imaginary line. Here you can also see the cerebellum and the brainstem. Here you can see the brainstem consists of pons which is present rostrally and medulla present caudally, which further down continues as spinal cord. The yellow structures emerging from the brainstem are your cranial nerves. You can also see the optic chiasma and the olfactory bulb on higher level. Here you can see a fully developed ventricular system in adult brain and how it appears from lateral side and from top view. So before we begin the journey of understanding the brain and its function, it is imperative to know different planes and some sections of the brain. And this is very basic, and please make sure you understand this without any confusion. So if we consider a plane which is passing through the center of the body and dividing it into left and right. So here we have the structures which are closer to this plane, which are called medially located, and the structures which are present away from this plane are called as laterally located. So there is a median plane, towards it are medial, and away from it are lateral structures. Similarly, the structures which are present towards the front are called as anterior, and towards the back are called as posterior structures. We study all the three-dimensional structure along its three axes, that is x, y and z axis. Similarly, we study the brain by taking its slices or sections in three directions, and this system is used for imaging like CT scan and MRI, and also in a dissection hall. So let's study what are these three planes along which we take the sections. First, we can take the sections in a plane which is parallel to the ground, like this. So these are called as axial sections. So one which are parallel to the ground are called as axial sections. Then the ones which are parallel to this midline, which are going from right to left, are called as sagittal sections. And the ones which are going from front to back are called as coronal section. So like this is axial, like this is your sagittal, and one which are going from front to back are called as coronal sections of brain. So in this video we have seen the basic anatomy and development of the brain. We have seen about its covering, the meninges and the ventricular system, and we have seen how the brain looks from the different angles and in different sections. I hope you have become acquainted to this amazing organ and be ready to study about its function in the next video. Please like this video and subscribe to our channel to keep receiving similar content in future. And do comment about what you like and disliked about this video so we can update our methods